Great. Hello, everyone. My name is Natalia Prado, and I'm a conservation biologist for the Smithsonian Center for Species Survival and Center for Conservation Genomics. I will be moderating our last session, and our first speaker up is Arsh Suri. Can you go sharing your screen, please, while I introduce you? So Arsh is a 10th grade student from the Chattahoochee High School in Georgia and intern at SCBI. Arsh, can you correct if I said that wrong, please?
No, it's fine. Hello, everyone. My name is Arsh Suri. I'm from Chattahoochee High School. And this summer, with my mentors, we conducted research and developed a domestic dog ovarian tissue co-culture in a microfluidic system. So what exactly is a culture? Well, culturing animal tissue is a process of growing and developing cells outside of the animal's body. And this means that we can collect tissue and culture from anywhere and then culture them in the lab and grow them in the lab. And depending on what tissues you culture, you can impact a variety of fields. So to maximize this growth, we want to mimic the tissue's original environment, because that's where their original home is, and that's where they're happy and grow best. You can see a sample tissue culture on the right. This tissue is basically submerged in media or nutrients and hormones. So in our research, we're culturing tissue harvested from ovaries, an organ which produces eggs for fertilization. And culturing tissue from the ovary has direct applications to animal conservation, gene therapy, and drug toxicity, and more. But how exactly does it apply to conservation as a whole? Well, let's say we have an endangered species. We first want to collect ovarian tissue from that endangered species. And then in an ideal situation, we want to culture that tissue in an ovarian tissue culture system. From this, we can then collect and mature oocytes, or developing eggs. Then, through in vitro fertilization, we can have pups form, which will actually increase the number of endangered species in that population. However, this full process has only been established for a few specific mammals, let alone endangered species we can't test on. So how do dogs come into the picture? Well, by further developing this process for the domestic dog, it can better apply to wolves or other endangered canine species, resulting in their numbers going up. So this summer, we focused our efforts on improving the efficiency of ovarian tissue culture, because currently there is no successful consistent ovarian culture system for dogs or other canines. And as you can see, this culture is a very important step for this process. And to do this, we want to maximize the growth of these tissues. So we must mimic this ovarian environment, or the tissue's original home. To do this, we first have to understand the ovary, or the ovarian environment. So follicles are essentially small sacs containing and developing an egg, or an oocyte. And the ovary has thousands of these follicles, and it's maturing and developing these eggs, and therefore these follicles. And actually, you can see on here the way these follicles develop. It starts off very small and then grows in size and ovulates. So here you can see a primordial stage follicle, where the oocyte is very small and there's little to no helper cells. And as these granulosa cells, which help provide growth factors and help them in general, attached to this oocyte, this layer forms, and now transitions to the primary stage follicle. Then as more of these hang on and connect, it can transition to the secondary stage follicle. Then this cavity, this antral cavity, will start to develop. And eventually, the antral follicle, or the final stage of development, will ovulate, meaning it'll release this egg. But only a few follicles will ever ovulate naturally. For example, in human females, only 300 to 400 eggs are ovulated, and the rest sit there. That means almost all follicles are unused in this ovary. And through innovation in tissue culture, we can then develop and fertilize these unused follicles and eggs. For example, if an animal were to die unexpectedly or in some way be unable to mate, we can use culture to develop these untapped follicles and increase their numbers. But what exactly causes follicle development? How does this process start? Well, follicle development is spurred on by hormones. But many cells outside of the follicle cells, outside these granulosa cells, play a role. For example, vascular cells can help transfer essential nutrients for development. But what are some ways people currently culture ovarian tissue? So one method is called an agarose gel culture, and you can see a diagram on the left. We first collect and divide this ovarian tissue and place it on top of this agarose gel. And you can think of this gel as a sort of a gelatinous jello, and it's partially submerged in this culture media, and this culture media are the nutrients and hormones the tissue might need. And the agarose essentially acts like a sponge, sucking up the nutrients in the media and giving it indirectly to tissue. But what's special about this culture? Why is it widely used? And that's because it forms an air-liquid interface. The tissue is both exposed to the air and liquid. You can see that here, the tissue interacts freely with the air and then also interacts the liquid through this interface, this agarose interface. This is important because it goes back to the point where this better mimics the ovarian environment and the tissues can grow more. But another technology people use to culture ovarian tissue is microfluidics. And you can think of this technology that is essentially uses these very tiny pipes to transfer nutrients to cells. You can actually see these pipes on the left. On the scale of millimeters, these guys are still very small. So our culture media will first flow through this small pipe and into the channel. Then our tissue will be placed in this channel and the media will be flowed through it. And as you may know, all living things produce waste, including these tissues. So the waste produced and all the other fluid will be flowed out and then cycled through again. But let's compare these two different culture methods. Well, first, agarose gel culture has huge advantages with its air-liquid interface. It better mimics this ovarian environment. However, has little to no automation, meaning a person has to individually manage the different aspects of media, and the waste is not removed or cycled. You can see that if the tissue produces waste, there's no way for it to be cycled or removed. And this doesn't really work on a large scale. However, with microfluidics, it is easily automatable, and this waste is cycled. However, it is very expensive. It requires a lot of specialized equipment, like a cleaning room, and specialized equipment to create these chambers. So it's very costly. So what we wanted to do is create a solution which will be the best of both worlds. It'll be less expensive, meaning less specialized equipment needed. It'll be able to cycle this with tissue produce waste. And it'll have this air-liquid interface, which will replicate the ovarian environment better. To first address expenses, we use 3D printers, and 3D printers are a very accessible technology, which allows one to fabricate any sorts of 3D geometry using a wide range of materials. And by using these 3D printers, we made molds. And these molds can then be filled with a PDMS. And this is a silicone gel, which will then serve as a vessel chip we flow through, the chamber we flow tissue through. And you can see a picture of this mold on the right, a picture of what we use. So after printing this, we would then fill it up with PDMS and let it cure. But how do we establish an air-liquid interface in a microfluidic system? Well, we made a dual chamber tip, meaning this top chamber will be housing the tissue, and it'll be directly exposed to the air through these inlets on the left and right. And on the bottom chamber, we will flow this media or the liquid aspect of the liquid interface from pumps. And as our interface, we'll have this membrane, this yellow membrane, which will be a fine porous membrane in between both these chambers. And it'll help function in the same way that agarose did in the agarose gel culture, a sponge for these nutrients. And this will better simulate the ovarian environment, and the tissue would interact through these cellular membranes. And in our system, the media, which contains all these nutrients or hormones, can then freely flow through the membrane. But how do we make this better? How do we even better simulate the ovarian environment further than an air-liquid interface? Well, as we said, other cells outside of the follicle, like vascular cells, play an essential role in development. So we decided to co-culture or also grow these vascular cells with the tissue. And these vascular cells could aid with nutrient transfer like it does in the ovary. And these cells can also provide all these growth factors. So how do we co-culture these cells? Well, we attach them to the bottom of that nice yellow porous membrane, and so that it'll be directly exposed to all these nutrients and fluid flow. But how do we decide the shape of the chamber? How do we decide the rate of fluid flow? Well, first, we can use fluid simulations, and we can generate this diagram. And in this diagram, you can essentially see the different paths a fluid can take in our chamber, in our bottom chamber. And these simulations are important because our vascular cells we're using in a co-culture can be damaged by the media's vorticity and velocity. And vorticity is basically the speed at which it rolls around. And in this diagram, where it's more red, it has increased vorticity. And where it's more blue, it has decreased vorticity. And you can see that only in the beginning do you see these slight hints of red or high vorticity. And in the center, it's very blue and has low vorticity. And it's exactly what we want. We don't want these vascular cells in the middle, which are attached to the membrane in the middle chamber, to be damaged by this media flow of vorticity. So how will we experiment using these cultures? Well, first, we'll collect ovarian tissue samples from dog 1 and dog 2, and this will be used for a culture. Then we fabricate our microfluidic chips, and we'll first set up 4 sample groups for both individuals. A first chip with microfluidic cells, with cells, and another microfluidic chip without cells. And the other 2 sample groups are going to be agarose gel cultures. And we'll first do an agarose gel culture with cells, and these cells will be in the medium themselves, and an agarose culture without cells. And by culturing these, or growing and developing them for a week, and then doing histology on them, or staining and letting us observe how these follicles grew, we can measure the follicular density and the dead zone percentage. Essentially, we count each follicle stage, and we measure the total area, and we can measure the density per millimeter of each follicle stage. But what exactly is a dead zone we're measuring? Well, a dead zone looks like this very white, palish area in the center of the tissue. And it's caused by nutrients being unable to flow to the center of the tissue in culture. And so by measuring this area of this very pale section and calculating a percentage of the total area of the tissue, we can see how much of this tissue is a dead zone. And this can serve as another factor to see how successful our culture is. Here you can see results from our data. On the y-axis, you can see the follicular density per millimeter in each follicle stage. For example, blue is for primordial stage follicles, and red is for transitional stage follicles for both individuals. We also measure fresh tissues, follicular density. And as you can see in dog 1, there's substantially higher follicular density. And in the agarose culture with cells, it has substantially higher follicular density. And you can see the same effect happening in the chip cultures. The chip with cells has substantially higher follicular density. In dog 2, you can better see the losses that culture can bring. We count the fresh follicles because they're a good metric on how we can measure follicular density, because all cultures will have eventually a drawback. So by measuring this, we can see that the chip with cells, the culture with cells, has a higher follicular density. But this data is not the final part. We only did 2 individuals. So results are still under construction. We're still building this up. And we can do this by having more individuals in part of our process. Here, you can also see the results from our dead zone. And on the y-axis, you can see the dead zone percentage over total area for both dog 1 and dog 2. And we count these dead zones for each culture, with these error bars being the plus or minus standard deviation. As you might notice, the fresh part of the tissue has little to no dead zone. And that's because these dead zones come from these cultures. It comes from the lack of nutrients being able to enter the middle. However, you can see in dog 2 that these dead zones are mostly present in the cultures with cells. And in dog 1, that both agarose cultures have similar dead zone percentages. So what can we conclude from this data? Well, first, we can conclude that successful and competitive ovarian tissue microfluidic cultures can be constructed using 3D printing and PDMS, and this minimizes its equipment and costs. These inexpensive cultures provide even more benefit, as you can then rapidly prototype them with little to no cost and experiment them with flexibility. You can also see that microfluidic chips with a co-culture of uterine vein cells seems to improve this primordial follicular density in a 1-week culture, meaning that the presence of the cells in the chip we developed is higher and could be more successful and be a competitive culture method. But we also noticed that the presence of these cells introduce these dead zones, these areas of dead tissue. And we think that's most likely due to these cells intaking nutrients. So what are these next steps? What can we do in the future? Well, like we said, our results are still under construction. We still need to elaborate on these trends by increasing the number of dog individuals. We can also increase culture time period. By culturing these tissue for longer, we can see how they interact, how the cells might interact on a longer time frame. We could also experiment with flow rates and volumes to combat the cell nutrient uptake caused by these different vascular cells. So that's it for my presentation. Thank you all for listening. I want to also give special thanks to my mentors, and Dr. Nagashima and Dr. Songsassen, my parents, and my teachers. And I'll be now happy to take any questions.
Thank you so much, Arsh. So I think to stay on time, we will move on, but Arsh will be monitoring the Q&A throughout the session. So if you have any questions for him, please just drop them down there and he'll be sure to answer for you.
Yeah.
Okay, so our next speaker is Madeline Becker. Madeline, can you please go uploading your, sharing your screen? So Madeline is with the Center for